Hello, in this video we will discuss about the history of viral discovery or virus discovery. So first of all, we should need to understand about the virus. And the virus is basically is come from the Latin word venom. Venom or poisoning, it means the toxic. So it can be defined as non-cellular, it means not cellular, non-cellular infectious entities which contain RNA, ribonucleic acid, or DNA encapsulated or encased in protein coat and produce only in living cell. So it means it reproduce only in living cell. It means the outer environment for the virus is inactive and non-living character, while when infect to the cell of the host, for example the human, which that is cell infect by the specific host. So the all virus are the specific for the specific host. For example, the HIV virus is the specific for the T lymphocyte because the T lymphocyte contain a spike protein receptor. So in this way, CD4 receptor, for example, it will be infected by the spike protein via a spike protein receptor, which that is present in the host cell. So this is the number one thing. And in this way, the inactive will become active when move inside. So in this way, it will be active and it is host 1 and it can be infected by host to host. So it means, for example, the sexual contact through HIV virus transmitting from one person to another through a sexual contact is important. HPV as well as HIV, so the hepatitis, etc. So on the other hand, here is you can see, and next we will discuss about the Louis Pasteur. That is a first work about the virus. So he did that about 50 years before developed electron microscope. The existence viruses was demonstrated as a large group of acellular disease causing agents composed of nucleic acid, for example DNA or RNA, which that is the genetic material, and encapsulated in protein coat by Louis Pasteur in 1884, which that is discussed about that, and a Mayer in 1886 during their structure study mosaic disease of tobacco plant. So it is also recognized. On the other hand, the next one was that the 1892, that is known as the Lewanovsky, 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 D. Lewanovsky. So that is a difficult name. So the prepared and extracted, he prepared the and extracted from tobacco plant that were infected from mosaic disease. So when the extracted was passed through a filterable to prevent the passage of bacteria. So the bacteria will not filter while the filtrate was able to infect healthy tobacco leaves. As a result of this experiment, these disease-causing agents were named filterable viruses. It means it can be filtered. So in this way you can see, and I am repeating again for understanding as well. So the prepared and extracted from tobacco plant that were infected from mosaic disease, and when the extracted was passed through a filterable to prevent the passage of bacteria, the filterable was able to infect healthy tobacco leaves again. As a result of this experiment shows that these disease causing agents were named filterable viruses filterable. Why? Because it is very minute as compared to the bacteria, but in this way we are using the filter paper, for example, for understanding. The filter paper is prevent the filtration of the bacteria, but the other smaller, because the bacteria is bigger than the virus. So in this way the those microorganism and the Lewinsky, which that is again put to the tobacco plant to create the again disease. So in this way the filterable things which that will disease So in this way you can see, and in this way basically the filtrable viruses is filtrable and cause the disease. So he uh, he, uh, he was not he was not able to see, but he was explaining about that. So in 1898, M. Nick established that viruses possess the property of reproduction, replication that is common to all living things, and he further established that these disease-causing filtrable viruses can multiply in the host cells, and it's required host for replication. So he was explaining about that in 1898. Bejernik, which that is, he was explaining about that a virus can replicate into the host only, and the outer environment for the virus is non-living. So here is another the later on the F Ward and F D Harley, which that is independently work, and that some viruses he was saying that some viruses infect bacteria, and they termed as viruses bacteriophages, and eater of bacteria are simply phages in 1935. And it is the W. Stanley which shows that tobacco mosaic virus or plant virus, and he crystallized the protein after that. So in this way the discovery will complete is here, you can see. And this was the history of virus, and next video we will discuss about the structure and function of the virus. So that was the overview of the history of virus.